Well, hello, ladies and gents. Hope you're doing well today. So in this lecture, we're going to do two quick parts on this. We're going to look at the notion of osmoregulation, the ability to control the movement of water through tissues of the body, because being able to do so allows you to sort of have opportunities to go new places. So, what happens is that organisms are able to control solute concentration or osmolarity, a measure of how concentrated stuff is, like per liter. Because if you can control the osmolarity or the solute concentration of tissues, then you can control the movement of water. Now, what's the value of being able to control the movement of water through tissues? It allows organisms to live in new and different places. Think about yourself. You have trillions of cells in your body all bathed in water, right? So life exists in water. But where are you? You're on land. So we are terrestrial organisms that can live outside of water, but we only do that because we can control the movement of water in and out of the cells of our body. So by being able to osmoregulate, by being able to control the movement of our water through our bodies, we can live in new places. All right. So osmoregulation is really about two things. One is maintaining homeostasis. You got to maintain constant levels of certain ions and nutrients and oxygen and stuff like that in the blood. And the other thing is excretion, because the breakdown of molecules through metabolic reactions is going to produce some waste products. So if you have trillions of cells producing waste, you're going to have some toxic blood. So you have to be able to get that stuff out, but also maintain the stuff you need. So how are you going to do that? Well, what does the work in the body? Proteins. Proteins do the work. So there are going to be special pumps and channels along the way that deal with the breakdown of these protein products so that we can get rid of the waste but maintain homeostasis. All right, so there are two ways to sort of deal with osmolarity. One is to be what's called an osmoconformer. Now, osmoconformers live exclusively in marine environments, and their tissues have an osmolarity that is the same as the surrounding environment. An example of this is sponges. Sponges, the osmolarity of their tissue is the same as the seawater around them. They expend no energy with trying to deal with that. And that is fine for a sponge because it lives almost its entire existence anchored to a particular location. Now, osmoregulators, they have tissues in place to try and deal with differences in osmolarity. So they'll have osmolarity levels that differ from their surroundings, us being an example of that, right? And again, the huge benefit is it opens up niche opportunities. So now organisms, multicellular organisms, can live on land. So we can live on land out of water, but be made out of cells bathed in water because we can control the movement of solutes and water. So the big benefit is for us to be an osmoregulator is we can live outside of the water. The downside is we have to expend a lot of energy and materials to regulate that. Osmoconformers, their sort of downside is they have limits to their environments where they can go, but they do have the ability to expend quite a bit less energy. Let me pause for a second. Excuse me. Here we're going to talk about osmoregulation as it compares to humans and the cute little kangaroo mouse that can spring around or bounce around. Now, kangaroo mice will live in hot, arid environments, but they can survive on very little water a day. Now, if we look at the comparisons here between these pie charts, humans, a lot of the fluid we produce or consume every day is lost in urine, and then we exhale quite a bit. For the kangaroo mouse, it produces quite a bit less urine because it has the capacity to sort of get rid of waste but retain water. So they've evolved to retain that water. Okay. Now, I would encourage you to sort of be familiar with the differences between freshwater and marine fish, because these examples have been described in problems on the AP exam before. Now let's consider the existence of a saltwater fish. Saltwater fish, what type of environment do they live in? It's a saltwater environment, right? So that would be a hypertonic solution. And if it's a hypertonic solution, that means there's a higher solute concentration outside the cells of the fish. Well, if there's a high solute concentration, that means there's a low water potential. And since water flows from high to low potential, water would tend to flow out of the tissues of saltwater fish. So what that means is they will constantly be drinking seawater, and they produce very little highly concentrated urine. They tend to store some of the waste products for metabolism in their tissues. That's why, like a shark, if people eat shark, it has to be prepared in a certain way because there's so much ammonia in the tissues of the shark that it's toxic. Again, saltwater fish live in hypertonic environments, so they would tend to lose water from their cells. So they're constantly drinking and producing very, very little urine. Now let's contrast this with freshwater fish. Freshwater fish, there's not a lot of solute in the surrounding water in a lake or a pond. So because they live in a freshwater environment, that is a hypotonic environment. In a hypotonic environment, that means there's a higher solute concentration inside of cells. And if there's a higher concentration of solute inside of cells, that means there's a lower water potential inside of cells. So water would have a tendency to flow into the cells of the freshwater fish. Boom, it creates lysing problems. So freshwater fish will drink very little water, and they produce gobs and gobs of very dilute urine. So they're just urinating all the time. So if you have some freshwater fish in a tank at home, they're constantly urinating to try and excrete that water out of their bodies. All right, a fun example is marine iguanas. Now, these reptiles live on the Galapagos Islands, and what they do is they swim down into the seawater and eat plant material down on the rocks beneath the surface of the water. So they're consuming a lot of salt water as they feed. So let's look at how they deal with that. So what these marine iguanas do is the salt that they consume, they actually concentrate it in glands in their nostrils, and then they squirt out like really salty water to secrete the salt that they have consumed swimming around in the water. So again, they have a really salty nasal spray. There's your slow-mo. Okay. So, imagine what Darwin could have thought seeing something like that. So again, marine iguanas is a fun example of how to osmoregulate. All right, now an issue that all these organisms are going to run into is that when you perform all these chemical reactions, inevitably you're going to break down proteins and nucleic acids. Now, proteins and nucleic acids both contain nitrogenous compounds or nitrogen containing compounds, and some of those can be fairly toxic. Again, ammonia is fairly toxic because it'll interrupt the production of ATP, so you don't want that. For us, we can take ammonia and immediately convert it to urea, so we can store urea in our bodies longer. And so whenever you have these nitrogenous wastes, you got to get them out of the body, right? So what we need to do is develop a way to get rid of nitrogenous waste, but retain water. Again, we're land-dwelling organisms, and so we need to have a capacity to get rid of our nitrogenous waste, but retain as much water as possible. So in the next clip, we're going to look at how our kidneys can do that for us. All right, thank you much.